Alright, so this is part 3 of 3. Now that we have a host, we've got E2 bacteria, let's find something that eats it. Let's find a phage that infects E2. And so we went back out to the Penobscot and we collected samples and we brought them back to the lab, and then we mixed them with pure E2 to see if we could find phages that infect it. And we did! So we found actually several phages that can infect E2. And again, statistically, they're new because we've only discovered 0.0002% of all the phages out there. So this was really exciting. We purified these phages in the lab, and then I took the students over to the University of Maine, Orono, and the students learned how to prepare the samples for electron microscopy, and then the students actually took the image themselves. They learned how to use the microscope, and the students produced all of these images that you can see here. So we've got 5 phages here. We've got phage A, C, D, E, F. If you're wondering what happened to B, well, we started to isolate it and it didn't grow out. So we just have, rather than relabel our phages, and that would have been confusing with our freezer stocks, we just let B go. So we've got phage A here, and we started characterizing this one in detail first, just because it was the first one. And then we've still got to characterize phage C, D, E, and F. And phage A, you can see it's got the head here, it's got the tail, and then really hard to see, but it would have legs here too. And we measured the length of the tail and the width of the heads, and we can see that they all different. They all have different sizes. And we looked at several images, and all of us measured these independently to make sure that we had a good representative sample. What's really the most exciting one, so we might skip over C, D, and F next and go straight to E. You notice E doesn't actually have a long tail, which is really, really cool. Most phages that people have studied and isolated do have this morphology or this type of structure. So here we've got one with just a tiny little, almost looks like a hook there. And so this type of phage is more rare. You don't discover this one quite as often. So we might move to this one next. This is a really cool image here that one of the students took. In the center, you have just a bit of flotsam, just debris. This is a piece of a cell. And these phages have made a mistake, and they think that this piece of a cell is actually a bacterial cell, and they've tried to inject their DNA into it. And so you can see this cell still has a long tail. These, as it injects the DNA into the piece of debris, the tail contracts. So this is really like pushing the plunger on a syringe. It contracts. You can see these 3 with a contracted tail, and this one where it's still elongated. So this is phage A, this is phage A, this is the one that we started to characterize in the lab first. So we grow it in bacterial cells, and what does it look like? And I'm going to try to zoom, and if I can make this bigger. So on this plate in the background, all this cloudiness, that's bacteria growing. You put the bacteria on the plate first, and you overlay a mixture that has phage in it. And if a phage lands, then it starts to eat the bacteria, bacteriophage, or it lice the bacteria. And what it is that it creates an area of clearing. So all these little spots here, those are areas of clearing or plaques where a virus is replicating. And when you isolate these, you stick a little toothpick into the plaque, and then you transfer it to a test tube, and you kind of shake it up, and then you take that, and then you put it on a plate again, and you try to see if all the plaques have the same shape and size and clarity, and then you do it again and again and again to get that really pure sample, and then you can study it. And these plaques are clear. That means the virus starts to replicate right away. Some phages can actually integrate into the genome of their host, and that's called lysogeny. And when phages do that, they're not all synchronized anymore. They're not all immediately get in, kill, go to the next cell, get in, kill. They get staggered, and so some of the viruses will enter and they'll go into lysogeny. So the phage genome will integrate into the bacterial genome, and some will kill the bacterial cell right away. And so you get these sort of cloudy plaques. So you don't see as much bacterial cell death, but our phage, phage A, is lytic, so it's killing the cells right away. We found that it needs active cells to replicate. This was expected. Let's see. Yep. So if we look at this figure here, on the x-axis we've got time, and on the y-axis we've got absorbance, or the optical density. So you take the test tube, you shine a laser through it. The more light that gets through, the less stuff there's in that tube to bounce the light around. So if the tube is full of bacteria, then the laser gets bounced around with all the bacteria and less light gets through to the other side. There's a detector. If there's no bacteria in the tube, then all the light gets through. And we can use this to measure how effective the virus is at killing the bacteria. If the virus is really effective, then you'd expect there to be less bacteria in the tube, and therefore the absorbance would go down. If there's lots of bacteria in the tube, the absorbance goes up. So we've got here in the green, we've got two lines here. So we've got our stationary control, and we've got the phage that's replicating in stationary bacteria. And stationary bacteria are bacteria that aren't actively replicating. And you can see there's really no change here. There's lots of bacteria in the tube, so that light is getting bounced around a lot. So the absorbance is pretty high. The bacteria are absorbing the light. You're not getting good transmission. The transmission is low, the absorbance is high. So, and you're not really seeing, and these lines almost overlay, you're not really seeing a big difference here. So the phage is not doing a good job of killing these bacteria. In these two lines here, we've got exponential control in purple. So this is the bacteria by itself, and the bacteria start off at a low concentration, so a low absorbance, and as the bacteria grow, the absorbance increases. So more bacteria in the tube, the laser gets bounced around more, the absorbance goes up. Now, in this line here in blue, we've got the virus mixed with the bacteria. So initially, the cells start to grow, but then the virus catches up, and you start to see the absorbance go back down. So the bacteria grows much better in our actively replicating cells than in the stationary cells. We wanted to know how quickly does the virus replicate. So you start off with a small amount of bacteria and a small amount of virus, and you mix them together, and then you take samples every few minutes, and you plate those out, and then you count the number of plaques. And this gives you an idea of, you can do a quick calculation, you can figure out how fast the virus is replicating. And on this graph, we've got time again on the x-axis, and we've got titer. This is the amount of phage on the y-axis. And this is the experiment done twice. Again, this is preliminary, we need to do it again. But we can see the virus is growing, and then you can take the number of viruses at the end, divided by the number of viruses you started with, and the number of bacteria that you put in there, and you can figure out how many viruses are getting reproduced in every infection. And so with this, we calculated a burst size of about 434, which, to be honest, is a little bit higher than we expected, so we've got to do this a couple more times, and I expect the number to go down a little bit. But this is how we're able to see how quickly the virus replicates in culture. And then we look at how stable it is in the environment, and this was kind of exciting. We can see the virus is actually really stable over a wide range of pHs. So on the x-axis we've got pH, and on the y-axis we've got the amount of virus, the amount of phage. And we take the virus and we normalize this to pH 7, so that's 100%. So we incubate the virus in media that's at pH 7, and then we take some virus and we incubate it in media that's at these different pHs, and then we put it on a plate and we let it grow, and we count again how many plaques we have the next day, so we can see how stable it is. And if it's stable, so then we're close to 100 here, and then you can see it drops off. So even down to pH 4, actually, we've only lost half of our phage, and down at pH 11, you still got some detectable phage here. This peak, we've got to do this a couple more times to see if this peak is real, but we're pretty close to 100% across a wide range of pH. And then we lose it pretty quickly with these steep drop-offs. So what we think is happening here is that virus is pretty stable until you get to those extremes of pHs that denatures the proteins, and then the virus just literally falls apart. And then we also looked at how stable is the virus with temperature. So on the x-axis we've got time, and then on the y-axis again we have the amount of virus. And we took the virus, again we normalized this to 4 degrees. At 4 degrees we know the virus is pretty stable. And then we took some virus and we put it in a test tube and we held it at different temperatures for different amounts of time. And then you plate it out and then you count the number of plaques. So you can look at the stability of the virus. And if we're looking at temperatures here, 60 degrees, that's these red triangles. Everything below that, so even 50 degrees, 37, pretty stable. We lose very, very little virus. But when you get to 60 degrees, it drops really dramatically. So this is a log scale. We're at less than 1% here. And then at 70 degrees, you have pretty much nothing. Since this graph, we've done a couple more experiments, and we found about 55 degrees right in the middle. Gives us a result kind of where we'd expect in the middle here. Again, what we think is happening is the virus is a pretty stable structure, and then if you just bring it, it's stable, and you heat it a little too much, and you denature the proteins in the virus, and it literally just falls apart, and you lose your stability. So this is the basic characterization of a brand new virus. We're hoping to repeat some of these experiments, get a little bit more reliability, and we're hoping to publish this next year. And I want to emphasize that all of these experiments were done by students. The last bit of data, so again, we're going to repeat these a couple times. We're going to try to isolate the genome. That's what we were working on in the lab actually right before spring break, and then we haven't been back since, but we were trying to isolate the genome of the virus, and so we'll send that genome off to get sequenced, and the sequence of the genome plus these data hopefully next year we'll be publishing this. Well, I hope this was interesting to you guys, and I hope you're doing okay at home, hopefully comfortable, maybe in your pajamas, learning a little bit about some of the research that we're doing at Hassan. Thanks for your attention.